Good afternoon, everybody, and welcome to today's How Tuesday webinar, which is dedicated to coastal and ocean acidification data. Janet, can you give me a quick thumbs up that you can hear me okay, loud and clear? Awesome. All right. My name is Carl Villacoba, and I am the Communications Director for Monmouth University's Urban Coast Institute, which is a very snow-covered place located about a mile from the shore in New Jersey. And I work as the day-to-day project manager for the Mid-Atlantic Ocean Data Portal. I'll be your moderator for today. And so today's webinar, working with the Mid-Atlantic Coastal Acidification Network, or MACAN, we recently added a collection of almost 250 new maps to the portal showing monthly surface readings over the course of 5 years for four parameters that are instrumental for determining acidification levels and events in coastal and ocean waters. Over the last decade or so, we've worked with MACAN to publish a lot of other acidification data on the portal as well. So if you browse our water quality theme, you'll find several map layers showing sites where acidification indicators are currently or have been monitored in the recent past. MACAN has also released infographics showing thresholds for pH and aragonite saturation, where various mid-Atlantic species could experience mortality and or stunted growth due to acidic conditions. So in today's webinar, you'll hear more about this data and MACAN's research and educational outreach on coastal and ocean acidification from Janet Reimer, who is the co-coordinator of MACAN and a member of the portal's technical team. Following Janet's presentation, we'll open the floor to a Q&A. And given that so many offices in our region are still closed due to yesterday's blizzard, we're expecting a smaller crowd than we would have thought, which means we can also keep this a little more informal. So feel free to hit the raise your hand button at any time if you have an immediate question, and we can probably field that right then. I am recording this session for those who can't be here, and it will be posted on the portal's blog and webinars page by tomorrow, if not today, and I can also share it with Janet's group to get it on the MACAN website as well if they like. And with that, I will turn it over to Janet to get us started.
Thanks, Carl, and thanks for the great introduction. I think I can just skip everything. No, I'm kidding. So I'm going to give you a little background on MACAN today, and then these digital tools that we've put together mostly with decision makers in mind, but that's a very loose term. So we'll talk a little bit more about that. So the coastal acidification networks are a federally funded initiative, and there's 7 of them across the United States. They were initially established in 2009 and have gained funding and momentum through different acts of Congress and executive orders. So we're very pleased that such attention has been given to acidification, but it's also important to note that our funding is always contingent on the need for the research. And given that coastal acidification is a water quality issue, whereas ocean acidification is more of a climate issue, we've been tending to focus a little bit more on the coastal aspect of it recently. So I'm not going to bore you with the congressional history, but just know that most recently, the 4AM Act established the NOAA's Ocean Acidification Program. Then in 2016, MACAN was formed, and it's the third youngest actually third youngest of all of the coastal acidification networks, with the Caribbean being the most recent addition. Then in 2022, we got a large boost in funding and appropriations through the CHIPS and Science Act. And then most recently, MACAN and the other coastal acidification networks have been funded through NOAA's Ocean Acidification Program core funding. But then again, MACAN also had a large chunk of funding from bipartisan infrastructure law from 2023 through 2026. So when we talk about acidification, it can be a relative term. But what's really important to remember is that when we say acidification in a coastal or an oceanographic context, what we really mean is a decrease in situ or in water pH that is detrimental to species and habitats that is not naturally occurring. And that's an important distinction because there are natural changes in the acidity or the alkalinity, which is the base properties of seawater that do occur naturally through different processes. And I'll walk you through a couple of those. But what's really important to just think about is that ocean pH water should be between 8.1 and 8.2. So that's where you get this open ocean line, which is very similar to the pH of your blood. Then we get into the coastal zone and we get a little bit more acidic, but again, that's a relative term, with estuarine acidity usually ranging between 7.1 to 7.9. So it depends on condition. And then when you get into your riverine water, rivers are the most acidic. And in this case, sometimes river water or fresh water can get down into the low sevens and the high sixes. So again, it's all relative, but this scale is important because it's also a logarithmic scale. So that 0.1 is actually much more significant than just, 0.1 if you're thinking about inches or centimeters. So when we talk about coastal acidification, what we're really talking about are these in-water processes that contribute to decreased pH, or in this case, an increase in the free hydrogen ions that are dissolved in water. Now, I'm not going to get into all of the chemistry today, but that's really the most chemical that you have to understand. And those hydrogen ions get released naturally by various different processes, including the bacterial decomposition of organic matter. And the more organic matter that you have in estuaries, rivers, and coastal zones, you have even more of that organic matter remineralization, which releases even more inorganic nutrients, which can fuel further production of organic matter. So there's kind of this positive feedback that occurs. So what we really want to do is make sure that we're not putting excess nutrients into the water to spur that organic matter growth. Another point source can be sewage outfall or septic seepage. You can also have agricultural runoff, which will bring additional nutrients to fuel that organic matter growth. Again, rivers are inherently lower in pH, so they are another source of low pH in the coastal zone. And then with the construction of many impervious surfaces, such as parking lots and roads, you don't have that natural filtration of the water through the water table and into rivers in the coastal zone. So without that natural removal of nutrients, you automatically get more nutrients that spur the organic matter that then enhance, again, the decomposition and the release of inorganics, and especially CO2. And then once you have more CO2, you end up with more free hydrogen ions. So like I said, I'm not getting into the full chemistry, but these are all processes that contribute to the acidification that we find in some of these areas. And again, these can be either long-term, which is a climate issue, which we find mostly in the open ocean, or they can be kind of these sporadic events that occur in the coastal zone. So one of these things that we really want to understand is, okay, if pH is decreasing, is it decreasing enough to have an impact on biology? And if it is, how do we know that that is an actual negative impact to biology? So these are some questions that MACAN and the other coastal acidification networks seek to answer through the research and data synthesis that we do. So we'll get back to that in a second. So if we want to think about how do we track the locations that could be at risk for acidification? And when we say acidification, it could either be in the open ocean, that long-term slow decline, or in the coastal zone and inner estuaries, large fluctuations that could be just these acidic events. But are the acidic events detrimental to species, or have species kind of evolved to withstand some of these low pH events? Well, the answer is yes and no. Some species are more adaptive than others, and some species just naturally have a greater capacity to withstand these negative impacts from large fluctuations in ocean pH or coastal pH. So MACAN is seeking to put together information on the water chemistry, which for acidification is called carbonate chemistry, with biological sensitivities to those low pH events. Now, we put that all together, we put it on a map, and then we can provide that information to resource managers, scientists, educators, restoration planners, and aquaculture and fisheries locations. Because remember that when we use our coastal zones, we don't just use them for one type of resource. There are many different resources and ecosystem services that our coastal zones provide. So we need to understand a wide range of biological conditions, chemical conditions, and how that's impacting the habitat overall. So one of the potential biological impacts, and this is important to understand because low pH affects different species differently and to a greater or lesser extent. So this is just two examples of laboratory studies that were used to understand how high or low pH due to high CO2 can impact species. So on my left, which might be your left as well, we have a 4x4 graphic of a little creature called a pteropod. And pteropods are a zooplankton that are really important in the oceanic food webs. And so these particular examples are pteropods exposed to high CO2 conditions over a period of time. So in the top left is the intact shell, and then you can see that the shells kind of deteriorate in quality as time goes on. And I do apologize. Usually I have a graphic that has the number of days, but I believe the most deteriorated shell was up to 39 days of high CO2 conditions, which generated the low pH conditions. And then on my right, you have some eastern oyster that was different animals that were exposed to different pH levels for the same amount of time. I believe it was 30 days. And you can just see the lower the pH, the shell quality does seem to change. So you have the kind of the wearing away of that nice, solid, hard brown substance. And I'm not a biologist, so forgive me, I don't remember what it's called, to the more delicate white layer underneath. So we do have plenty of laboratory studies that show us that there are negative impacts to species and that each species responds uniquely. So when we think about carbonate chemistry, we can collect these water we can collect information from the water in various different ways. And I'm going to show you why this is important in one of our portal layers as I get to it. So we can collect underway or moored continuous CO2 concentrations through different methodologies, all measuring what we call the partial pressure of carbon dioxide, which is the carbon dioxide that's dissolved in seawater samples. Then in the coastal zone, we can even, wheel carts out onto docks to grab samples. Those are generally for dissolved inorganic carbon and total alkalinity, which is a different type of sample from the dissolved CO2. Rutgers University is leading a glider program on the East Coast that collects pH. Sorry, their program collects pH from these wonderful gliders, and they send them out all year long. And we're really excited because we're starting to add some of that glider data into the portal database. And I'll talk a little bit more about that. And then on cruises, we can collect water samples. We generally can collect them through the water column, depending on conditions that day on the cruise, but we can take deep water samples up to several thousand meters. So we can get a really clear picture of what carbonate chemistry looks like through the water column, but those samples are few and far between because those research cruises don't happen often enough. When we measure carbonate chemistry, we're measuring, like I said, for dissolved inorganic carbon total alkalinity, the partial pressure of CO2, and sometimes we can directly measure pH. Of these four different parameters, we need at least two of them to be able to calculate the other parameters, which is great because that means you don't need to collect everything everywhere. So with that in mind, we started putting together some of our portal layers. And before I jump into the actual layers, I also want to just talk about a little bit of the project background. So in 2026, we started compiling, or sorry, 2016. In 2016, we started compiling information to produce what we're calling monitoring maps. And those maps kind of just established where and when and what is being measured for acidification within the Mid-Atlantic region. Then in 2017, MACAN hosted a workshop to identify monitoring gaps over space and time. In 2019, MACAN published a paper in Estuarine, Coastal, and Shelf Science that gave recommendations on where to monitor in light of the gaps that were identified from the 2017 workshop. In 2021, we started putting together what we call our assets inventory, and that was a huge outreach effort that Kirsten Wakefield, who was a former MACAN Co-Coordinator, did pick up in 2022 and took that on, literally reaching out to all of the different researchers and state agencies and local planning offices throughout New York, New Jersey, Maryland, Delaware, and Virginia to understand who was monitoring in what exact location. Then MACAN hired a fellow in 2023 who started compiling not only all of that, she compiled all that information together, but she started sorting it based on what's being measured at specific sites, and at those specific sites, how many parameters of the four of dissolved inorganic carbon, total alkalinity, pH, and the partial pressure of CO2, how many of those were being sampled. Then we had a contractor take on part of this project in 2024. Teresa Schwimmer started to calculate the different monthly averages, and she also put together a huge database of the biological responses in species in the Mid-Atlantic. And so that's where we started to get this intersection of biological impacts with the carbonate chemistry. And so the portal layers that are up there right now were the work of Teresa, and they are averaged into the gridded slider layers. And for the different sliders, so that means you can see the monthly progressions over the time frame of 2018 to 2022 of how pH, the partial pressure of carbon dioxide, hydrogen ion concentration, and then this other parameter called aragonite saturation, which we'll talk about in just a second. We can see how all of those have been fit together to understand how acidification changes on a month-to-month basis. Then pairing that with biological impacts, we can start to understand which locations and species are going to be at the greatest risk. So then I said I was going to tell you a little bit about the aragonite saturation state. So this aragonite saturation it's a variable that we can calculate from either any combination of the four different carbonate chemistry variables. And it's an indicator that tells us how likely shell is to form or not form in a region based on the acidification level, or in some cases, basification, but we are not the Mid-Atlantic basification network. We're just the acidification network. So we're not going to talk about alkalinity and basification today. So I'm going to pause there before we start getting into the data layers, and just to make sure that I answer some questions that I do see in the chat. So we have one question that says, how do you distinguish between natural fluctuations and anthropomorphic driven changes? There are some statistical methods that we can use to tease that out. We can look at proxies going back. So we have CO2 information for the atmosphere going back to the most recent ice age. So we know that in general, that carbon dioxide is increasing in the atmosphere. And if you're really interested in how some of these statistical methods are applied, I can throw some papers into the chat at the end, but it's quite a different and in-depth topic. But we use statistics. That's the short answer. And what parameters do we collect on moored buoys? It depends on the buoy, but in general, there's carbon dioxide detection sensors that we can use. There's different pH sensors that we can use. I know that there's been some beta testing of alkalinity sensors, but those have not come to this country yet, and I haven't seen any of the peer-reviewed literature yet, so I'm pretty sure they're still in development. How far back does the data go? In the Mid-Atlantic, we have a fairly continuous data set that goes back to 2018, but it depends on the site as well. There's pH data from state agencies that goes back sometimes into the 1960s. However, the data quality has changed over the years, so you really have to be careful about which data you're using. That's not to say that it isn't reliable. It's just that with changes in technology, some of the data it's not a one-to-one comparison. And I would encourage you to visit the MACAN website for more information on that, and some of the MACAN specific webinars that do focus a little bit more heavily on the science behind acidification. It's not consistent across states, the data collection. It depends on the state agencies as well. So I'm going to leave it at the short answer of just saying it's not consistent. And I think I probably touched a little bit on how consistent sampling techniques are, given that there's 4 different parameters, and in some cases there's 3 to 4 different methods for analyzing water samples. While the methods may not be entirely consistent, we all try to focus on getting internal consistency and validation through I guess you could say through collaborations, but that doesn't always work out, which again is why I say there is little consistency, but it's not for a lack of effort. There's a lot that goes into it, and funding is one of the major factors. So I hope that answers those questions to a point where just kind of have a level of understanding of why this kind of data synthesis project is not only such a huge undertaking and has taken us 10 years and several different stages. So, all right, I'm going to get back to the maps. So I did screenshots because I just wanted to make sure that I didn't have any problems with clicking over to the website and the layers potentially loading at a slow speed. So this particular map is it's under our, as Carl mentioned before, all of the acidification data is under the water quality drop down. And you can see that up at the top here, I did select all acidification monitoring sites. So what the legend would tell you, because you can't see the legend and which data you're selecting at the same time, but what the legend would tell you is that there are 4 different types of samples that were collected here. So you have yellow, green, actually, I'm sorry, it's more than 4, but the circles that you see kind of as discrete circles are one time, like it's a discrete sample. So it's a sample that is taken and then you move along. Everything that's connected, those are cruise tracks. So some of those cruise tracks are gliders, some of them are from shipboard measurements. So, and then the different colors also indicate the different parameter that was collected. So I'm just going to say, for example, this red line is one of the East Coast Ocean Acidification Cruises, or ECOA cruises, and it is likely a dissolved inorganic carbon or a total alkalinity sample that was taken there. So that's one of the examples of the maps, is that you can see where all of the different acidification monitoring sites are. And and these are also historical and current. So some of them could be an ongoing sampling regime, or they could have been sampled once and that was it. This is an example of total alkalinity and dissolved inorganic carbon sites. So the total alkalinity is the yellow and the dissolved inorganic carbon is the green. And again, these can be either from one cruise, multiple cruises. This is just an example of the maps that show you where, but not a when. Then of the 4 parameters, and this is why I mentioned earlier that this was important, is that if you have only one parameter that was actually measured in the water, that's it. You just have that one parameter. You can't calculate anything else. So those are the light blue triangles. And you'll notice one of the great things that we do have is we have it all the way up into the Hudson River. And several other rivers as well. You can see around the Chesapeake Bay watershed, we have lots of measurements up into that one, or up into the rivers. This map doesn't tell you which parameter it is, but it just tells us that there's only one parameter measured. Then the purple is where there are 2 parameters measured. The red is where there are 3, and the yellow is where there's all 4. So as I said, as long as you have 2 parameters, you can calculate the other ones, which is going to give you information about why acidification is changing, not just that it's changing. And then if you have 3 or 4, the more accurate those calculations can be. So if you have 2, you can get a good idea. 3, better, and 4 is the best. So if you have 4 parameters, you're golden. I think maybe that's why we chose the yellow color. And it gives you the most information about why acidification is changing. Then we start to move into some of the sliders. And I have a couple of different stages of this. So for just this example, under the acidification monthly sliders drop down, this one is specifically pH. And this pH is monthly from 2018 to 2022. And you take a look at this map and you think to yourself, well, I saw all of this data from before. Why am I only see a couple of locations? Well, it's because, like I said, not all of the samples were taken at the same time. So we have a lot of time mismatch, which becomes an issue. However, one of the great things that we can do from this is that we can take all of that data and we average it into these gridded cells so that we can, in a sense, extrapolate and have better coverage and understanding of why pH changes, where it changes, and when it changes. So we are still working on projects that will expand our prediction abilities, but for right now, we're so excited that these data are available for use. So it is that I used the same layer and I just clicked over so that you can see where it's sliding. So for example, this is September from 2022. And again, this is pH. And we used a colorblind safe color scheme. So it's pinks and light greens so that it stands out from the blue background. And the lighter the pink into the green color, which you can see just a little bit east of Baltimore, the greener it is, the more acidic it is. And that's exactly what we would expect to see, the lighter colors and the more acidic conditions inshore versus out on the shelf, where you would assume that you'd have an ocean pH of around 8 to 8.2. So then what I'm going to do is I'm going to take you over to our biological impacts graphics. So you can find these not on the portal, but we will soon have a link from the portal over to the MACAN website from the Ocean Stories page. And so we are midacan.org. And up at the top of our homepage, you'll see a drop-down menu for species impacts. And then from species impacts, you would click on biological impacts from acidification. And here I am taking you over to the website right now. So from species impacts, up at the top, you click on biological impacts from acidification. There's a lot of information on here about how to understand and use these graphics and how they were put together. But today I'm going to take you directly to view the graphics under this button here for View Biological Impacts Graphics and walk you through one of the ones on pH. So we also have this information for aragonite saturation, but today I just want to focus on pH, and maybe at a subsequent webinar, we can dive deeper into aragonite saturation state. But pH is a much more universal understanding of an acidification level versus aragonite saturation. So we're interested in understanding mostly mid-Atlantic species. Some of these species are found in the Northeast and do have a southern extent as well. But one of the things we're interested in is at what pH range does mortality occur? And not just for adult species, but we're interested in larval, juvenile, adult, any species state or any life stage. And it is kind of a trend that larval stages are more susceptible to acidification than adult stages. So all of this information is compiled from 81, I believe it was 81 different peer-reviewed papers, and we do have references for those at the bottom of the graphics. So this red line indicates where lethal effects have been detected or below their point or range at where the pH condition begins to cause mortality. So that means if you have a body of water that has a pH of 7.9 and then it starts decreasing and it gets to a pH of 7.7, we would expect to see larval death of lobster, of the American lobster. And then anything below that, again, we would expect either mortality or just a lack of spawning to begin with. And just kind of for reference, we put in the average estuarine pH range so you can understand that, yes, these species likely do experience some natural low pH, but in laboratory studies, any extended period of time at a 7.7 pH or lower, there has mortality has been recorded. So we have American lobster, Atlantic cod, Atlantic silverside, bay scallops, blue crab, blue mussel. And you can see for the blue mussel, we have embryo stage, larval stage, and juvenile stage. And we also indicate where these life stages are located. So for some species, like for blue mussels, for example, we would expect their larvae to exist in both estuaries and ocean. But however, this particular study that is being used as a reference was an ocean. It was simulating open ocean. So we have many different species. Most of them are economically important species in the mid-Atlantic, or they are the base of the food web of an economically important species. So that's what we have for that's what these graphics are. So the idea is to couple the graphics with the with the with the slider layers. So you could say, for example, I'm going to go back over to the presentation. So let's say we saw that spiny lobster begins to have, I'm sorry, not spiny lobster, American lobster, that American lobsters begin to show mortality at a pH of 7.7. And just for the sake of reference, let's say that sorry, even though like the range of American lobster is a little bit farther north than what I have, it was the first example on the graphic. So let's say that right here in the lower Chesapeake Bay, you have pH that is not conducive to American lobsters. That let's say it's a pH of 7.1. So if you're a manager and you're looking to understand where you can implement some mitigation methods, you want to understand what's going on with the chemistry and then understand what you can do with the biology. You wouldn't want to time mismatch these things because then your restoration effort could be all for naught. So it's really important for managers to understand the timing of pH changes as well as what life stage and what biology is found at that location at that time. So these maps and the biological impact graphics are meant to be used together to help understand planners and resource managers and decision makers, help them understand how to manage acidification and when it's the most important to implement these management strategies and to also understand what species they might be targeting. So I believe that was really it as far as the slides that I have prepared. And we'd really love to take some of your questions, additional questions to help you be able to use these graphics to the best of your ability.
Thank you, Janet. That was really excellent. And kudos to you for being able to juggle all of that and read the questions in the chat and answer them when you did. So we're caught up there. And right now, I'll just open it up to anybody in the audience who would like to either leave one in the chat or just raise your hand. Maybe while people think about it, I'll throw it out myself. First, congratulations to MACAN, to all of you for what you've done here. It's a pretty big deal when I think back to 10 years ago when we had what was then one map, and it was an earlier version of what you showed on screen with that all acidification monitoring sites. And at the time, it had a lot less data too. And then over time, we wound up breaking that out into the by parameters to, you know, the the active sites and all of that to help people view those things individually. And one question we would always get in those early days was, all right, so those are the sites. Can we tell by these what the actual readings are? And we couldn't say. At the time, we could say no, but you can click on them. You can get a pop-up that would tell you who you can ask about that, so we can direct people to that. But now with these maps that you've put together, you actually can get those readings and see what's out there, which is a long way of getting to my question, which is so what's next? I know you do have some other related data coming, and maybe if you want to give people a little preview of what that is.
Yes. So we have two projects right now that are going on simultaneously, not exactly independently, but we hope that there will be some collaboration between the two. One of them is to take the biological response data from the biological impacts graphics and combine it with the carbonate chemistry data to come up with one single metric that would kind of give an understanding of at risk, highly at risk, or extremely at risk for acidification. And that would include an understanding of what's happening to the biology and what the chemistry is doing. It's harder than you would think because of the natural variability in the background that needs to be filtered out, because of changing climate conditions that need to be included, and because of all of the different processes, both biological and physical, that occur in coastal waters and open ocean. But I don't know that I would classify any of these samples really as strictly open ocean. Most of them are shelf and near shore and coastal. And then we have another project that is combining carbonate chemistry data and shellfish data. So I think we're looking at the eastern oyster and surf clam, which are two of the most economically important shellfish species on the East Coast. So we hope that by mid 2027, those projects will be complete with their new layers up on the portal.
Thank you for that. Who else has a question? Any questions? Also, if you have any questions about the portal in general, I am happy to answer those as well.
While folks are thinking about that, I will also say that MACAN will be hosting another webinar series that we're currently planning for the upcoming, I would say probably late spring, summer this year. Typically, it's been in the early spring into early summer, but we're pushing it back these days just because of all of the other work that we have going on. But you can sign up for the MACAN newsletter specifically from the MACAN website because we don't always overlap with the portal.
We got one question in the chat about whether there will be an e-certificate after this webinar. And Zyla, yes, you can just, I think you have my email address. You can just reach out to myself and or Janet, and we will take care of that for you happily. Greg, go ahead.
Hi, I'll just come off, Mike. So whenever there's some sort of a data portal that's available, the question of data quality always comes up. And so my question is this And I'm kind of throwing a bone to Janet on this question because we've had this conversation before, but it's worth talking about publicly. Is that, are these data portals are say all inclusive? In other words, like, do you have everything from like very, very high level data to say, citizen science collected data that may not have any sort of quality control applied to it, but the duty is for the person who's downloading the data or accessing the data to also do some sort of review of a metadata document or something like that. So I figured I would just bring that up for the group.
Thank you. And that's a really good point. I didn't talk about that. We have metadata for every single data point. Every point is cataloged through its respective data set. Um, the data sets are either already passed through their own peer review. Like, for example, some of the data is from the large scale NOAA cruises, some of it's USGS data, some of it is state agency data. And as I pointed out before, not all methods are the same, but we have information on the method in which on the method of which it was collected. And we don't currently have citizen science data in the acidification world, at least not here in the Mid-Atlantic. There are some initiatives in other regions, but to my knowledge, they're not publicly available through through our types of data portals. The portal is essentially federally funded, so we do have to follow, and we do follow data QA/QC metadata protocols. So you can rest assured that any data that you're getting through us has been through appropriate processes. In addition, I just want to add, the portal does not house the data itself. So we provide these synthesis maps, but there are links to where you can retrieve any and all of the carbonate chemistry data from. And because it's from a whole bunch of different sources, we hold in-house one large, very, very large data set of all of it compiled together, but that's not for public release. That's for our use because we feel that the way that we use it may be different. And so we encourage anyone that's interested in obtaining the data to get it directly from the source.
Hey, any other questions? Very well. I think we will stop here, and I'll just thank everybody again for joining us and thank Janet for this great presentation. And mention again that we will have this online. I'll try and get it up by the end of the day, but certainly by tomorrow at the latest. So thanks again all for joining, and hope you all are doing okay with the snow, and it will melt as soon as possible, and we'll get to spring. Thanks again, and have a great finish to your day.